Cementum. Cementum is a specialized calcified substance covering the root of a tooth. The cementum is the part of the periodontium that attaches the teeth to the alveolar bone by anchoring the periodontal ligament. Structure. The cells of cementum are the entrapped cementoblasts, the cementocytes. Each cementocyte lies in its lacuna, similar to the pattern noted in bone. These lacunae also have canaliculi or canals. Unlike those in bone, however, these canals in cementum do not contain nerves, nor do they radiate outward. Instead, the canals are oriented toward the periodontal ligament and contain cementocytic processes that exist to diffuse nutrients from the ligament because it is vascularized. After the apposition of cementum in layers, the cementoblasts that do not become entrapped in cementum line up along the cemental surface along the length of the outer covering of the periodontal ligament. These cementoblasts can form subsequent layers of cementum if the tooth is injured. Sharpie fibers are part of the principal collagenous fibers of the periodontal ligament embedded in the cementum and alveolar bone to attach the tooth to the alveolus. If cementum can be observed on teeth, it can imply that the roots are exposed, showing that the clinical crown, the exposed part of the tooth, is bigger than the anatomical crown, the surface of the tooth covered by enamel. This is often due to gingival recession and may be an indication of periodontal disease. Cementoenamel junction. The cementum joins the enamel to form the cementoenamel junction CJ, which is referred to as the cervical line. 3 possible types of transitional interfaces may be present at the CJ. The traditional view was that certain interfaces dominated in certain oral cavities. The CJ may exhibit all of these interfaces in an individual's oral cavity, and there is even considerable variation when one tooth is traced circumferentially. Dentinocemental junction. When the cementoid reaches the full thickness needed, the cementoid surrounding the cementocytes becomes mineralized or matured and is then considered cementum. Because of the apposition of cementum over the dentin, the dentinocemental junction DCJ is formed. This interface is not as defined, either clinically or histologically, as that of the dentinoenamel junction DJ, given that cementum and dentin are of common embryological background, unlike that of enamel and dentin. The dentinocemental junction DCJ is a relatively smooth area in the permanent tooth, and attachment of cementum to the dentin is firm but not understood completely. Types. The different categories of cementum are based on the presence or absence of cementocytes, as well as whether the collagen fibers are extrinsic or intrinsic. It is thought that fibroblasts and some cementoblasts secrete extrinsic fibers, but only cementoblasts secrete intrinsic fibers. The extrinsic fibers within a cellular extrinsic fiber cementum travel perpendicular to the surface of the root and allow the tooth to attach to the alveolar bone by the periodontal ligament PDL continuous with the cementodentinal junction CDJ. A cellular cementum only contains extrinsic collagen fibers, whereas cellular cementum is quite thick and contains both extrinsic and intrinsic collagen fibers. The first cementum to be formed during tooth development is a cellular extrinsic fiber cementum. The acellular layer of cementum is living tissue that does not incorporate cells into its structure and usually predominates on the coronal half of the root. Cellular cementum occurs more frequently on the apical half. In summary, the main types of cementum are as follows: a cellular extrinsic fibers cementum EFC, cellular intrinsic fibers cementum CIFC, and mixed stratified cementum MSC, which displays both cellular and acellular cementum. Cellular cementum contains cells and is the medium of attachment of collagen fibers to the alveolar bone. It is also responsible for minor repair of any resorption by continued deposition to keep the attachment apparatus intact. A cellular cementum does not contain cells and has a main purpose of adaptive function. Composition. Cementum is slightly softer than dentin and consists of about 45% to 50% inorganic material hydroxylapatite by weight and 50% to 55% organic matter and water by weight. The organic portion is composed primarily of collagen and proteoglycans. Cementum is avascular, receiving its nutrition through its own embedded cells from the surrounding vascular periodontal ligament. The cementum is light yellow and slightly lighter in color than dentin. It has the highest fluoride content of all mineralized tissue. Cementum also is permeable to a variety of materials. It is formed continuously throughout life because a new layer of cementum is deposited to keep the attachment intact as the superficial layer of cementum ages. Cementum on the root ends surrounds the apical foramen and may extend slightly onto the inner wall of the pulp canal. Development. Cementum is secreted by cells called cementoblasts within the root of the tooth and is thickest at the root apex. These cementoblasts develop from undifferentiated mesenchymal cells in the connective tissue of the dental follicle or sac. Cementoblasts produces cementum in a rhythmic manner on intervals indicating periods of activity and period of rests called incremental lines of Salter. Incremental lines of Salter are the only incremental line in the tooth. Unlike in enamel, ameloblast incremental lines of Ritius and dentin odontoblasts incremental lines of von Ebner. The inorganic portion is much more than the organic portion, so when ameloblast and odontoblasts rest they leave a space for the organic portion and become hypocalcified. Unlike ameloblasts and odontoblasts, which leave no cellular bodies in their secreted products, during the later steps within the stage of apposition, many of the cementoblasts become entrapped by the cementum they produce, becoming cementocytes. Thus again, cementum is more similar to alveolar bone, with its osteoblasts becoming entrapped osteocytes. Cementum is capable of repairing itself to a limited degree, but not regenerate, and is not resorbed under normal conditions. Clinical significance. Some root resorption of the apical portion of the root may occur, however, if orthodontic pressures are excessive and movement is too fast. Some experts also agree on a third type of cementum, a fibrillar cementum, which sometimes extends onto the enamel of the tooth. The excessive buildup of cementum on the roots of a tooth is a pathological condition known as hypercementosis. Cementum thickness can increase on the root end to compensate for attritional wear of the occlusal/incisal surface and passive eruption of the tooth. When cementum is exposed through gingival recession, it quickly undergoes abrasion by mechanical friction because of its low mineral content and thinness. The exposure of the deeper dentin can lead to problems such as extrinsic staining and dentinal hypersensitivity. The incidence of cemental caries increases in older adults as gingival decline occurs from either trauma or periodontal illness. It is a chronic condition that forms a large, shallow lesion and slowly invades first the root's cementum and then dentin to cause a chronic infection of the pulp. Because dental pain is a late finding, many lesions are not detected early, resulting in restorative obstacles and increased tooth loss. Cementicles are small, spherical or ovoid calcified masses embedded within or attached to the cementum layer on the root surface of a tooth, or lying free within the periodontal ligament. Cemental spurs can be found at or near the CJ. These are symmetrical spheres of cementum attached to the cemental root surface, similar to enamel pearls. Cemental spurs result from irregular deposition of cementum on the root. They can present some clinical problems in differentiation from calculus and may be noted on radiographs, yet, because they are hard dental tissue, they are not easily removed and thus may also interfere with periodontal treatment. DNA studies. A 2010 archaeological study has found that cementum has 5 times the amount of mitochondrial DNA compared to dentin, which is commonly sampled. Teeth are increasingly utilized as a source of nuclear DNA to aid identification of human remains. DNA extraction and the results of genetic analysis from the tissue are extremely variable and to some extent unpredictable. However, the quantity of DNA available in dentin is affected by age and dental disease, whereas that in cementum is not.